Please join me in welcoming Megan Beaver.
Thank you very much, Judy, for that introduction. I'd also like to thank Pastor Todd and the chapel for setting the science forum up and inviting me to speak. I was very excited to get the invitation, so that was great. As Judy mentioned, I am the Director of Operations at the Akron Fossils and Science Center. Like she said, it's in Copley, about 20 minutes from here. We do a lot of fun stuff, so if you haven't visited, please do that. Especially in the summertime, it's loads of fun. We have a zip line. We do birthday parties, science classes, all kinds of stuff. So come out and see us. Every person that comes to the Akron Fossils and Science Center gets a guided tour. And on that tour, we basically present the information that the science forum that Judy and the chapel has put together. We present that in the tour. Is it logical to have a belief in God? When you're talking about science, a lot of times we hear that you can't believe in God with evolution, and science has proven evolution. We don't need God anymore. So we like to go through that and kind of explore, is that really true? Is it an option to exclude God when we're talking about science? Now, as a zoologist, of course, my area of study is animals, so that's my favorite part of the museum. I'm a little biased on that. But evolution kind of started by studying animal life. Charles Darwin's theory of natural selection came about by his visit to the Galapagos Island and looking at the variety in that area. So tonight I want to talk a little bit about natural selection and evolution. There's some confusion about those two phrases. Many times they're used interchangeably, so I'm going to clear up a couple of definitions, and then we're going to really delve into the meat of the matter and talk about the tree of life and genetics and see if the new studies in genetics are really supporting what we have been taught about natural selection and evolution. Now, as we discuss these, we're going to go through many different topics, especially genetics at the end with the new research. So let's go ahead and hit on it. I have a clicker, so we're going to do this the old-fashioned way. Now, natural selection, I have a technical definition up here, but basically natural selection is an observable process. We can see this happening, and it's basically just traits in a population becoming more or less common as those organisms in that population reproduce. So for example, say we have a population of frogs. Some have green skin, some have yellow skin. Okay? As they reproduce, you can have different colors come about. So those traits are going to become more or less common. You can have more green frogs or more yellow frogs, but as they reproduce, they're still going to be frogs. So that is a very important point to make. This is also the process where the phrase survival of the fittest came from. We've probably all heard that. Traits that are good for the population are selected for, those are kept in the population, and the bad traits are weeded out. Now, evolution, on the other hand, is the whole process. When you think about evolution, you think about chimps turning into humans and things of that nature. Natural selection doesn't do that. Natural selection acts on a population changing the traits, but we don't see those small changes becoming larger changes to do this. So the theory of evolution is basically it uses natural selection as a process, acting on random mutations in the genome to produce traits and to change that population. And then we result in biological diversity. Now, a random mutation, just for our purposes right now, we'll get into that a little bit later, but a random mutation is just a change in the DNA of an organism in that population, okay? Now this picture up here, the phylogenetic tree of life, this probably sums up the process of evolution the best. When you see this graphic, this really gives you an idea of what the theory of evolution is all about. Basically, it starts with that one cell at the bottom, a single cell that changed and evolved into all these organisms that we see at the top. Okay? So as you go up the tree, the organisms get more complex. The simpler organisms, or the less complex ones, are at the bottom. Now with this phylogenetic tree, you see that we have three distinct branches. One over here, you can see that plant life is represented. The one in the middle, we have arthropods, crustaceans, things like that. Can everybody hear me when I'm not right by the microphone? In the back? Okay. And then over here, this tree branch, this clade, as we call them, as a whole. Now, the tree of life idea was based on the morphology, or how these organisms appear physically. And we use taxonomy and classification to group these organisms in this layout. When you take a look at the clades, if you narrow in on a smaller section of that overall tree of life, you can take a look at some of the characteristics. Now, this is just showing turtles. We've got lizards, snakes, other reptiles, crocodiles, and then birds. And how this works is you take a characteristic that all of these animals share, and you group them by that characteristic. As you get more and more characteristics that you share, your group is going to become smaller and smaller and smaller as those traits are shared by just a few. Okay, so right here, feathers. Birds have feathers, the crocodiles, the lizards, and turtles, of course, don't. So you can move up the tree with these characteristics, grouping these into smaller and smaller groups. This is just another example. Over here we have the amniotic egg for reptiles, birds, and mammals, and then of course we've got the gizzard for just the crocodiles and the birds. So moving with those traits, you can group these smaller and smaller. Now this is a simplistic view of the vertebrate clade. So anything that has a backbone, we would put in this group. Now these dots right here, these are the nodes on the tree of life, or the phylogenetic tree. The nodes is where evolution is supposed to happen. That's where those characteristics come into play. So right here at the bottom, common vertebrate ancestor, common jawed ancestor, common tetrapod, which means 4 limbs, common amniote, so an amniotic sac in reproduction, and then you've got the mammals grouped up here together. Now these phylogenetic trees, like I said, were based on morphology and taxonomy, and also by looking at the fossil record. When we take a look at fossils, we can group those animals according to what class they belong in the phylogenetic trees and in taxonomy. Now these phylogenetic trees are allegedly supported by the fossil record. However, we don't find any of these common ancestors in the fossil record. No indisputable examples have been found at any nodes on the tree of life. Now every once in a while someone will come up with a fossil or organism that is hailed as a transition or the missing link. A lot of times we'll hear that. Just recently we had Ida was a type of lemur. They did finally classify it as a lemur, but that was supposed to fit in the ancestry of humans. When we take a look at these, however, we can always classify them in a group. An ancestor of these necessarily has to be different enough from the ancestors and different enough from the descendants to be a common link in that chain. So when we're able to classify all these into separate groups, it's counterintuitive. It really doesn't add up when we're talking about these nodes. Now one classic example of a missing link is Tiktaalik. Okay? And this is just showing in that circle the tetrapod group, so things with 4 legs. Tiktaalik was hailed as the transitional fossil or the missing link for all tetrapods, as fish evolved into amphibians and then into mammals and birds and everything with the 4 legs. So Tiktaalik is right up here. It is an extinct animal. Now these were found in Ellesmere Island in Canada in 2004. There were 3 specimens that were found. They were dated at 375 million years old and hailed as the ancestor of this group. This is what it looked like. Does anyone see any legs at all? There's no pelvis on the back end, and there are no limbs to be able to study. So why this was hailed as a transition for limbed creatures, scientists are still unsure. There's a lot of argument about this. Why was this allowed to get carried so far? It has since been classified as an extinct type of sarcopterygian fish, which is a lobed-fin fish. So it's not a tetrapod, it doesn't have legs, so this fell out of favor as an example. When I was in college, one of my professors had this on his background of his computer, and the caption said, Tiktaalik is a transitional fossil, so shut up. So he's been proven wrong, so it's somewhat amusing there. This is a transitional animal. Now, like I said, it's necessary for these animals to have traits that are different from their ancestors and different from their descendants. So it's counterintuitive to put this together when traits are so different. So until we find an animal like this, as a zoologist, I'm going to kind of hold off on agreeing with evolution and natural selection. Now, sometimes you might hear that, well, the fossil record might not be complete. There might still be stuff out there to be discovered. And we're making discoveries all the time, which is fantastic, but we're not discovering any new major groups of animals. And the fossil record is very complete. After over 200 years of scouring the earth, scientists have really filled up the fossil record. We're not finding, new major groups. We've got fossilized bacteria, fossilized leaves. So things that are very delicate are also being fossilized. So to say that the fossil record isn't preserving these things or it's not complete really doesn't hold up when you think about the evidence and the actual data that we see. What about punctuated equilibrium? Now, this is a rescuing device for the theory of evolution coming with natural selection. And what this theory discusses is it's trying to explain why we don't find these animals that should be at these nodes on the phylogenetic tree. This was developed in the 1970s by a couple of scientists, Niles Eldridge and Stephen J. Gould. And with this theory, what it says is that we take a look at the fossil record and we find a lot of stasis, which basically means no change. So they like to throw around the phrase evolutionary stasis. These animals that are going through evolution really aren't changing a lot. Now the phrase evolutionary stasis I find hilarious, and they use this a lot when I was doing my studies. Evolutionary stasis, the phrase literally means change over time with no change. And that's how they're explaining evolution to us. So take that how you will, but I think it's humorous. Now, punctuated equilibrium, like I said, it's explaining why we don't have these ancestors at the nodes. When you look at the fossil record, you find a lot of animals that are very similar to the animals of today. On the left, we have a fossilized bat, dated at 60 million years old. This is a bat, a contemporary bat, a little brown bat, and one of them, if you were to find a skeleton outside today of a little brown bat, that's what it would look like. I don't see a whole lot of change going on between these two. This is a fossil frog that was dated at 34 million years old. And when they took it out of the rock, it was still squishy. You can somewhat see the red over here on this side on the eye. So 34 million years old, and it was still rotting. So, and it looks like a bullfrog like you would see outside. So again, not a whole lot of change with these phylogenetic trees. Speaking of trees, we've got 3 leaves up here, and many of you could probably identify these instantly. Got an oak leaf over here. This would be a birch and an aspen on the right-hand side. So again, fossilized leaves, which leaves are very delicate, and they're being fossilized as well. So very good preservation in that fossil record. This gives you an idea of how punctuated equilibrium is a little bit different from gradual evolution, like we talked about earlier with that phylogenetic tree. When you look at this, as you go up the tree through time, you get these different changes, and they're supposed to be slow. With punctuated equilibrium, the changes happen too fast, so you don't see them as you go up the tree. These horizontal lines represent where evolution is happening, and because the change happens so fast, it's unlikely to be preserved in the fossil record. So basically what they're telling us is that the slow, gradual process of evolution is happening too fast for us to observe. So, my question to you is this a good theory? Is it a logical theory? The whole premise of this forum is faith in God logical. Let's take a look at this. Is this logical at all? Okay? So something to think about when you're talking about punctuated equilibrium, which I like to call punk-eq for short. It takes too long to say those words. But when you've got punk-eq and phyletic gradualism, neither one of these are really supported by the evidence that we see. Darwin's finches is a classic example of punctuated equilibrium, punk-eq. You probably have all heard of Darwin's finches. You've got a population of finches on the Galapagos Islands, and many of these finches have different shaped beaks. The beaks are shaped differently for how these animals live. So this guy in the middle has a nice thick beak for chopping open seeds. This guy over here is going to eat fruit, so he's got a nice thick example of evolution, this is an example of adaptive radiation, which is a process of natural selection. Traits becoming more or less common in an overall population. So you have different traits, but these are all finches. They're all the same animal. We're not having major changes in this population to change into an entirely different organism. Here's another example. Many of you saw the skull on the table as you walked in. That is from this animal up here at the top, Beaver, which is a type of extinct salamander. These guys could grow to be 12 feet long. The salamander on the bottom, this is Ohio's state amphibian, just like we have a state bird, which is the cardinal. We have a state amphibian, this is the spotted salamander. These grow to be about 8 inches. This is Disco, our salamander at the museum. So, gotta show off my babies. Again, dog breeds is another example of natural selection. You've got massive variety among the domestic dogs. Great Danes, Bernese Mountain Dogs, we've got French Bulldog here in the middle. Now, if you take 2 French Bulldogs and breed them over and over and over, are you ever going to be able to get a Great Dane out of those 2 French Bulldogs? No, of course not. These dogs all share a common ancestor because they're all the same species of dog. They're just different varieties, okay? So you can have different varieties of the same species, but these guys are never going to be able to reproduce and then turn into a different variety of dog because they've lost that genetic information. You're not going to breed 2 Great Danes to get a Chihuahua. It just doesn't work. You've got boundaries in those genetics, so it's not going to happen. So what we actually observe in the fossil record and in contemporary observations, we see groups of animals that have slight changes, but we don't see them with a common ancestor, because we don't have the evidence of that, which is what's shown at the bottom. So we don't have fossil evidence, we don't have observations that we're making today in the real world to support this at the bottom. This is what we see. Head back to that phylogenetic tree. Like I said, we don't have a lot of evidence in the fossil record. We don't see these major changes happening. But what about genetics? If these animals have been able to reproduce and change over time, there should be some evidence in the genetics of these animals to support that. And we're going to head there next. But at the bottom of that phylogenetic tree, remember we have that simple cell at the bottom. Let's take a look at a simple cell. This is an E. coli bacteria. It's a single-cell organism. Doesn't look like there's a whole lot going on here. Pretty simple when you take a look at it like this. We're going to zero in on a bit of this cell and take a look and see how simple these actually are. So we're going to zero in on the flagellum, which is basically the propeller of this bacteria. We're especially going to look at where it hooks into the cell body. So this is a blown up graphic of where that flagellum hooks into the cell, right up here. You've got the filament of the flagellum, so this is the part, this is the thread that hangs out the back. It hooks into the cell through a sleeve and a rod, and then you have the protein rings here at the bottom that hold that in, hold it together, okay? So again, pretty simple, doesn't look like there's a whole lot going on. The flagellum can spin and propel that bacteria through its substrate, so it's basically swimming with this flagellum of this bacteria. When the clutch is engaged, that flagellum will spin and move that bacteria through the substrate. When that clutch disengages, that flagellum will stop, the bacteria will remain motionless. Another cool thing that happens, this flagellum can then spin in the opposite direction to back that bacteria up through its substrate. This flagellum spins at over 100,000 rotations per minute. Yes. It disengages almost instantly and engages. Okay? Can happen in a fraction of a second to make this guy swim around. If anyone has ever seen these guys under a microscope, they're hilarious to watch. They're zooming all over the place because they can spin that flagellum so fast. Pretty simple cell, right? Doesn't look like there's a whole lot going on from this picture. You got some stuff in the middle, you got that nice thread hanging out the back, but it is very, very complex. And all of those pieces have to fit together and operate properly in order to make this system work. Like I said, they're required, not just optional proteins kind of hanging out to see what they can do. So is it logical to think that this would have happened just by chance, which is what the theory of evolution requires? Think about this. Does it make sense? That's the decision that we have to make. Single cells are sophisticated, not simple. When you hear the phrase simple cell, it doesn't make a whole lot of sense. These are very, very complex, very sophisticated in what they do. Now, we have to go from a single cell to multicellular organisms. Human beings have trillions and trillions of cells in the body. Okay, we're not just single cell organisms. We've got a lot. So we have to go from that one single cell at the bottom of that phylogenetic tree to thousands of cells. And interestingly enough, we don't have any 2-cell organisms, 4-cell organisms, 128-cell organisms. We've got 1-cell organisms, or you have organisms with thousands of cells. How did we make that leap? Now sometimes you'll hear the example of desmids, which is a type of green algae. And many times you'll hear that these are 2-cell organisms. That is incorrect. They are 2 single-cell organisms cooperating together by a channel. They can share nutrients for a little bit, but they will eventually split off and go and do their own thing. Okay? It's like best friends that had an argument and separate. When we take a look at some small multicellular organisms, these guys are very complex as well. This graphic shows a rotifer, which is a type of protist. They're just small multicellular organisms. Again, these are fun to watch under the microscope. They're almost like kids in bumper cars. But you can see all these different pieces and parts that go together to make this rotifer work. These guys will spin, they'll jump around, they'll swim around. Very complex as well. This is a hydra, another protist, another small multicellular organism. These guys, both the rotifers and the hydra, can be found pretty much in any pond, water, or stream that you can find. These guys have tentacles at the top, which is what I want to focus on for a moment. When these are under the microscope and you touch those tentacles with a probe, those tentacles will suck in almost instantaneously. There's a signaling cascade that happens at that very first cell at the tip that sends a signal down into the hydra body to suck that tentacle in. And it is a very complex process. These are the different tissues that are present in the hydra. So you can see the epithelial cells, the skin basically, the gastrovascular cells in the middle, basically the stomach, the gut of this animal. So that signal will travel from the tip of that tentacle all the way down into this area to get sucked in. And this is also how they eat. When they sting a certain type of prey, they'll stick onto it with their tentacles and pull it into the mouth, which is right up here at the top. And it'll go into the gut and they will digest their food. So again, pretty small, but sophisticated. Now, like I said, when we're talking about the phylogenetic trees and how these animals are supposed to evolve, we have to make a leap somehow from a 1-cell organism to multicellular organisms. We don't really know how that happened, but there should be some clues in the DNA. So that's where we're going to head next. We're going to talk a little bit about genetics, and I want to present to you some new research that's happening to see which idea is supported. Is it evolution or intelligent design? This is the structure of DNA. been twisted. The sugar phosphate groups are on the rails of the ladder, so this blue ribbon here in this graphic. And then the nucleotide bases make up the rungs of the ladder. Now the nucleotide bases, there's 4 of them, and they're pictured over here: adenine, thymine, guanine, and cytosine. These guys will pair up across those rungs in a certain fashion. Adenine always bonds to thymine, and guanine always bonds to cytosine. Okay? You can't have adenine bonding to cytosine and thymine bonding to guanine. They don't fit. So they have to bond in this fashion. When you look at DNA, especially in humans, it's very complex. This is the rope or the strand of DNA with all those letters, all those nucleotide bases lined up. The DNA will fold into what's called a chromosome. Humans have 46 chromosomes in their somatic cells, their body cells, 23 in reproductive cells. Those fit in the nucleus, the center of the body. When you read the DNA, there's about 3 billion subunits of DNA, so those bases, those letters on that strand. That equates to approximately 30,000 genes, which makes up the traits. So your eye color, your hair color, how big your nose is, things like that. These make up proteins, which determine those traits, determine the functions in the body. So again, the genetic code, nucleotide bases, they are paired up 3 at a time, or grouped 3 at a time. That codes for a specific amino acid. The amino acids line up in a chain to make the protein, which does the function in the body. I've got an animation that'll show you how this works. The sound was kind of iffy a little bit ago, so we'll see if it actually runs.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a 2-part chemical manufacturing machine called the ribosome. While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins. Using the instructions encoded on the messenger RNA as a template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain. As this chain exits the ribosome, it is met by chaperones which prevent premature folding, while escorting the protein to a barrel-shaped machine called a chaperonin. This machine helps fold the protein into the precise shape required to perform its function. Although it is unclear how the chaperonin achieves this, we do know that accurate folding is essential in order for the protein to accomplish its intended function. Once the protein is complete, it is released into the cytoplasm to do its job.
Look at how complex that folding was. And like she said, that folding is essential. It has to be folded in that proper fashion in order for it to do its job in the cell. Now what happens in that process if there's an error, if there's a mistake? That's what we would call a mutation in that DNA. So as that DNA is unwound and it's read by those proteins to make a copy, sometimes that copy has an error. For example, if you take a picture and have it photocopied, you're going to lose some of the resolution. If you take that copy of that photograph and copy it, you're going to lose some of that resolution again. As you copy over and over, sometimes those errors, those And this is what we're going to talk about next. Now mutations, we always hear that those help evolution along. Random mutations acted on by natural selection will produce a result, and the desired result of evolution is biological diversity. But does that actually happen? Mutations will do one of three things, sometimes more than one of these three things. They remove information from the genetic code, they rearrange information from that genetic code, or they ruin that information. Okay? Now sometimes we hear about beneficial mutations and how sometimes there are good things that happen with a mutation. Now, sometimes that's true. We do have something good that can happen, but not all the time. Now, when you take a look at these two sentences, they're identical, except for one letter. But by changing that one letter, you change pretty much the entire meaning of that sentence. If you put a cat in a bag, you read the sentence, the cat is in a bag, you've got a picture in your mind of what that looks like. Now, if we change that one letter, the C to a B, and the bat is in the bag, that changes your mental picture. It changes the information that's relayed by that sentence. The same thing happens with the genetic code. If there is a change in that genetic code, you will have a change in the information that is relayed to the cell. Now, if you let the cat out of the bag, you'll have a reaction. If you let a bat out of a bag, I guarantee you'll have a different reaction. Although I like bats too, so I wouldn't be bothered. But again, sometimes you hear about mutations that are beneficial, that help an organism, but no example of a beneficial mutation has been found that does not also have a negative effect. So you can have a gain of function, but you also have to have a loss of function as well. The common example that's talked about is sickle cell anemia, which is a disease of the red blood cells in humans. And this is what it looks like. Usually, the red blood cells are round, that's when they're healthy, and they've got a little bit of a divot in there to carry oxygen through the bloodstream. The sickle cells are kind of flat and squishy. They don't have that nice rounded shape. So they cannot carry oxygen as well as the red blood cells, as the round red blood cells. Now this is showing the sickle cell trait, where there's a mixture of red blood cells and sickle cells. Sickle cell disease, all you have would be those sickle cells. And death is the reaction of that disease. Because the blood sticks together, it can't carry the oxygen, so this is a problem. The sickle cell trait is beneficial in the fact that people that have that trait, have this mixture, are not susceptible to malaria. So that's always hailed as a good mutation. These people don't get malaria. However, if this person were to marry someone with this trait and have children, chances are one of their children would have the sickle cell disease, and that child would die. So natural selection would weed this out. It's not a beneficial mutation when we're talking about evolution. Now another mutation that's brought up recently is a mutation of the CCR5 gene, which makes people resistant to HIV and henceforth AIDS. However, it was recently discovered, I believe last year, that this mutation also causes liver damage. So yes, you won't get AIDS, but you'll die of liver damage. Which would you rather? I don't know. But it's always a toss-up. Now when we take a look at these cells, they are very, very complex. The information to build a cell comes from that DNA, like we said. And it also has ways to protect itself against those errors in that DNA. There are proteins that their specific job is to repair the cell or repair the DNA. So there's basically a backup system built in to the cell to make it healthy. Now sometimes there will be an error or a mutation that is so bad that the cell is not going to recover. So it undergoes what is called programmed cell death, or apoptosis. And that's what's pictured in this graphic here. If there's a mutation or an error that is so bad, that cell will commit suicide, basically, okay? It'll start to shrink and degrade and basically eat itself, tear itself into pieces, and then the body will devour that cell and essentially destroy it. So there are backup systems to these mutations if you do have things that are bad that happen. This is a photograph of human skin cells. You can see the healthy cells right here. These large, round, healthy-looking cells with the nucleus in the middle. You also can see some of these cells are kind of squished up. This one up here is essentially dead. Your skin regenerates all the time. You have new skin cells being prepared all the time, and your old skin cells will flake off. Now, most of the time you don't notice it, but has anybody in here ever had a bad sunburn and you get that flaky skin? Those cells are dead and they're flaking off because of that radiation from the sun. So you can see this happening. As cells reproduce, like I said, they will pass on information and pass on that genetic code. Now, sometimes there's errors in that code from the copies, but we're going to take a look at how these cells reproduce. This process on the left is called mitosis, and this is what happens in your basic body cells, your skin cells, your blood cells, your muscle cells. The process on the right is meiosis, and this happens in your reproductive cells. And I'll tell you why there's a difference here in a second. We're going to go through each of these. In mitosis, the one over here, this cell, each of these pieces in color are chromosomes. That's where your DNA is. When the cell is ready to divide, that DNA will copy itself, okay, and then it will line up and split. So each cell, each daughter cell is what they're called, will have the same DNA and the same cell structure. In meiosis, it's slightly different. This happens in the reproductive cells, and this is where we get our variety. This is why you don't look exactly like your parent. Your DNA is grouped up here. When it copies, it'll line up and split a little bit different from how it happens over here. Recombination happens, which basically what that is you'll have crossing over of two of those chromosomes. One chromosome will give another chromosome information, and that chromosome will swap. So there's crossing over, there's a swap of genetic information. This is where we get variety. For example, eye color. My mother has brown eyes, my dad has blue. I have brown eyes because of the recombination that happened in here. Brown is dominant to blue, so that's what shows up. This is where that happens. Now when this splits, you'll have the two cells will have slightly different information from each other, and it will also be different from the parent cell. In mitosis, they're all identical. This cell is identical to this one, and both of these are identical to the one at the top. So meiosis is where we get a lot of that variation. These two will split again with slightly different information, and this is where we get half the number of chromosomes. So like I said earlier, in your body cells you have 46 chromosomes, in your reproductive cells you have 23. When reproduction happens, 23 chromosomes from the mother and 23 chromosomes from the father will unite, and that's where you get even more crossing over and variation. Twins are a little bit different. They will look identical to each other sometimes, but not to the parents. This is just a better picture of the cell dividing, and these are just the different phases. And you can see what's happening. The DNA is lining up to split, and then the cell will split, and then each cell will have its own DNA and its own cellular structure. This is an example of real cells. The last one was a model. These are epithelial cells from a kangaroo rat kidney. These are under a fluorescent microscope. This is where I want to direct your attention. Cytokinesis is the last stage of cell division, and that's where the cell will split into two. Right over there. So you've got one cell that is split into two identical cells. These are somatic cells, so they're undergoing mitosis. So they will be identical. These are sand dollar cells. So a little bit different, it's not a mammal. But you can still see the same splitting. The two cells will split, so you have identical cells. This is how these guys grow and reproduce. But one thing that's very interesting is the DNA is not the only mechanism of inheritance. We always talk about DNA, and the genome is where all the information of an organism is stored. We're finding out that that's not correct. So when we're talking about mutations in the DNA driving evolution, we're we're really going to have to rethink that theory with the new information that's coming out in these genetic and cellular studies. The cell as a whole passes on information, not just the DNA. This is where we get different levels of information in the cell. So let me go back. You can see that graphic again. I want to make this point. This little blue spot is the DNA, but you can see all of this extra stuff in the cell, the fluid in the cell, the other machines that are operating in the cell to read the DNA. All of this is passed on to the offspring. Much information is included in that cellular structure, not just in the DNA. So we're going to explore this a little bit and talk about DNA or information in the cell. Now it's important to talk about information in an organized way when we're talking about biology. Many times you hear about information with computers and things like that. Biological information is a little bit different. There are 5 levels of biological information, and I'm briefly going to go through each one of these as we talk about DNA information and information in the cell. Statistics is basically the state of the nucleotide bases. So again, that A, T, C, and G, those 4 letters that make up the DNA sequence, where they are in that sequence is what we're talking about. They have to be in the right order in order for that DNA to make sense. Going back to that sentence, the cat is in the bag or the bat is in the bag. These have to be in the right order to make sense. They will then code for, they'll be grouped into 3 to make a codon. That codon is translated into an amino acid. We're just going a little more in depth with what we saw in that animation. In this case, AUG, those 3 nucleotide bases code for the amino acid methionine. And these will just line up to make that protein chain. That's all we're really talking about here. Syntax has to deal with the meaning of those codons. What exactly do they do? And it also has to deal with the starting and the stopping of reading these codons. So again, in that animation where that protein met a stop codon, that messenger RNA left that protein. So methionine is a start codon, and then we also have stop codon. So basically this is the punctuation of the genetic code, the punctuation of those sentences as they're read. So we start with a capital letter and end with an exclamation point, period, whatever. Pragmatics has to do with the practical application of the protein that comes from this DNA. So how it's folded. Remember I said that this has to be folded in a certain way. This is the chain of amino acids, which will become a protein. But it's not really a protein until it's folded. And like you saw in that animation, they have to be folded properly and be shaped correctly in order to do its function. And this is what a protein will do. Some proteins are enzymes. Those act as catalysts in a chemical reaction, and they will either join substrates together or break them apart in the cell. So to make products so your body can use, like energy and things like that. So these 2 substrates will enter that enzyme, a reaction will happen, and then this is the product. And that piece will go off into the cell and do a job. So it's basically like a key going into a lock. It has to fit. You can't have the wrong key to make this work. Alphabetics is the last dimension of information, and this has to deal with what the organism does as a whole. Back to the dogs example. We've got many different types of dogs. They're all the same species, just different varieties, and again, they're reproducing to pass on their traits. That organism as a whole has to do something. So it's going to reproduce and pass on those traits. Now, this information, the cellular reproduction, is an example of what we would call inverse causality. The present processes have some future goal. Okay? Now, when we talk about evolution, it's supposed to be a random process with no goals. It can't think. It's just something that happens in nature. But when we're talking about information that is present in the cell, it has a future goal. It has to do something. The DNA will provide variation, but the cell, its structures, and its physiology can serve the entire organism's purpose DNA operates. The cell itself is basically the book that the DNA text fits into. Okay? This might be kind of a gross picture. I'll skip through it real quick. But when you take a look at this, this is a raccoon fetus. So it's an unborn raccoon. That embryo, that fetus, is going to grow lungs. This raccoon in the womb does not need lungs. It has no use for them. If it had lungs in the womb, it would drown. But the raccoon outside its mom, once it's born, needs lungs. So basically, embryonic development, that information that grows in the organism during reproduction, has a future goal. It has to have all of these parts in order for that adult organism to operate. And again, I'm showing off my babies. This is banded. Yes, awesome. I will agree with that. Now, like we said, that cell encompasses the DNA and it operates as a whole. Information by itself is useless unless the cell knows how to use it. Evolution not only requires new information for new traits and new behaviors, things like that, but it also requires extra new information so the cell knows what to do with the new information it just gave. You have many levels of information that have to work in this cell to make these organisms. You can't just have a mutation and that's it. The cell has to know what to do with that mutation, that new trait that has come about. So you have multiple levels of information that's at stake here. So when you take a look at the phylogenetic tree of life, starting with that cell at the bottom, we know that those are not simple. We know now that they're very complex. That had to change dramatically. It had to have a physical effect on that cell for natural selection to act upon. Each node of the phylogenetic tree had to produce a physical effect on that organism for natural selection to act upon. And also, you have to have new information so that organism knows what to do with the new genetic information that it got from that mutation. The DNA and the cell and the organism as a whole has to know how to use all of this. That's why we see major groups of organisms with information. Humans will always produce humans, chimps will always produce chimps, salamanders will always produce salamanders. You have boundaries in that cell, in that organism, that limits the changes of the DNA that can happen. So, as we've gone through all of this, is it logical to think that all of this happened by chance, or that there's a designer out there that knows what he's doing and lets these organisms reproduce with very